population variation, uncertainty. It's good to see these words in the title. So thank you, Andrew.
Well, thank you very much, Richard, and it's a pleasure to be invited back here, and I really want to congratulate you and the organisers on this topic of uncertainty quantification. It's all the more of an honour for me to be speaking about it, since I really can only contribute U. I don't have much Q to contribute to UQ. But I will try, and the examples that I chose do have to do with uncertainty in data, and they kind of are loosely connected by the theme of ventricular remodeling. So I thought maybe I should start with some disclosures. The first one of which is that I always remember to put UQ in my grant proposals. I usually forget to put them in my papers. And I am one of those modelers who subscribes to George Box's aphorism that all models are wrong but some are useful. However, those of you who followed him and his ideas on this, he went on to conclude that the only good models were the most parsimonious and simple models, and that anyone who didn't use the simplest model was, you know, a scientific charlatan. But I don't actually go along with that, and it's because of the nature of biology more than the nature of modeling. Biological variability is a term that gets used in a lot of ways to mean a lot of things, and it can encompass a wide range of sources of data uncertainty, such as the effects of sex or diet or circadian rhythm or measurement error. One of my mentors, Henk de Kers, used to say that biological variability is just an excuse for experimental sloppiness, but it's not true. The experiment could be conducted perfectly, but the diet of the animal could have been a cause of biological variability that had nothing to do with the way the measurement was conducted. Spatiotemporal noise is not just a consequence of measurement uncertainty, but it's a real thing. Every cell expresses genes to different degrees, and that heterogeneity can often be a really important part of the tissue level regulation. And then, of course, there's genomic variation, and the thing is that the contribution of most of these sources of uncertainty to our conclusions about biological mechanism or the validity of a model are very poorly understood. So if I have a philosophy or a takeaway message from today, it's that a large part of UQ in cardiac modeling involves assessing how biological variability in its various forms affects our assessment of model usefulness. So the two examples that I'll talk about in a little more detail, one, our studies with Alistair Young, and who's now at King's College, I believe was here yesterday, and his former PhD student, Kat Gilbert at University of Auckland, his postdoc Arvin, my PhD student, Nick Forsch, and our clinical collaborators, Jim Perry and Sanjeet Hegde at Rady Children's Hospital, with Jeff Omans on congenital heart diseases and the Cardiac Atlas project. And the second problem that I'll talk about, where data uncertainty has been a big issue for us, is a collaboration with my former PhD student at UVA, Jeff Saucerman, his former PhD student Philip, and my former PhD student Kyle, my current PhD student Shu Lin, my lab manager Jennifer, Jeff Omans, and my collaborator, Keck Graduate Institute, Alex Zambon, and this is on network models of myocyte mechanosignaling. So the Cardiac Atlas project, for those of you who don't know Alistair or haven't heard about this project, derives shape models and statistical atlases from human cardiac magnetic resonance imaging exams. So starting with the 4D data sets, Alistair's group developed guide point modeling strategies for making shape models of the ventricles. Those parametric models, when you collect large enough numbers of them, can be used to derive principal modes of population variation in shape, which as a result of this dimensionality reduction can then lend itself to discovery through data sciences, or what used to be known as statistics. So they start by using a interactive guide point modeling strategy. Now can be done in biventricular way as well as in the left ventricle, where instead of segmenting the images, the contours are fit directly to the images with the aid of guide points representing anatomic landmarks that are specified by the user. And here's a biventricular model of a patient with repaired Tetrology of Fallot, which is something I'll talk a bit about. But let me start you with a bigger study that Alistair's group performed using nearly 2,000 asymptomatic patients from a large study called the MESA study, the multi-ethnic study of atherosclerosis. And so these are asymptomatic patients. These are patients who have no particular sign of heart disease. Here are the first three shape modes for that population computed for the biventricular atlas. Here are the first four shape modes being animated through plus or minus 5 standard deviations of the left ventricular atlas. And here is the percent of population variation described by each of these modes. And so you can see by the time you get to 10 modes, already 90% of the all of the shape variation of left ventricular shape across this asymptomatic population has actually been explained. So this is a really good way of taking these really large MRI volumes and reducing them to numbers that can be used to gain clinical and mechanistic insights. Because these come from 4D image sets, you also get, as a result, not just the end diastolic shape, but the end systolic shape and the shape changes during systole. And so there's different ways that can be analyzed. You can make a end systolic shape atlas, but you can also make a shape atlas of the difference of the displacements from end diastole to end systole. And what we see here is the first displacement mode shown as an end systolic shape against the end diastolic reference in the wireframe. And so you can see actually the prominent feature of the first mode of systolic wall motion here is the amount of longitudinal shortening. You can see that, you know, at one extreme there's a lot of longitudinal shortening, at the other there's none at all. You know, again, by the time we get to 10 modes, we're up to about 80% of the variation in systolic wall motion across this population has been explained. Okay. What if we want to take advantage of this insight in formulating models? How many principal modes do we need in order to capture the original shape to within a given accuracy? And here you can see that, you know, if all we use is the first mode, then the accuracy of the model, of the RMS error between the original data and the model, you get about 5.5 millimeters, compared with a voxel resolution about 1.5 millimeters. By the time we get down to 10, there's a wee bit of an inflection point here. We get this is right at the point of plus or minus 2 voxels. So there's still information to be gained by adding these additional components, but the rate at which the information is gained and the error is decreased is less. So, you know, right at about the point where we're reaching 80 to 90% of the variation explained is also the point at which we seem to get the best sort of return on investment for the fewest number of parameters to describe the shape to within 3 millimeters. Of course, these errors aren't uniformly distributed. You see they tend to be more prominent near the base than the apex, but by the time we get up to 10 modes, they're down at that 3 millimeter threshold. So Tetrology of Fallot is a common cyanotic congenital heart defect. It consists of 4 lesions. The first one is pulmonary stenosis. Okay? Second one is a ventricular septal defect. The third is right ventricular hypertrophy, which is really secondary to the increased pulmonary vascular resistance. And then finally, you have an overriding aorta, so that blood in the aorta comes from both ventricles. This is a cyanotic disease, and so these children, the babies that are born with this, have to undergo surgery typically within the first year of life to repair the pulmonary vessels and the pulmonary valve, close up the septal defect. But nevertheless, these patients live to adulthood, but they are nevertheless at risk of heart failure pulmonary regurgitation, right ventricular hypertrophy, and sudden cardiac death. Chronic pulmonary regurgitation is a common complication, and there's a question as to if and when there should be a pulmonary valve repair done in these patients when they're adults. The current guidelines for the timing of this pulmonary valve replacement, which nowadays is often being done by a percutaneous catheter procedure, are not very reliable for predicting whether and when the valve replacement will prevent subsequent adverse deterioration in right ventricular size and shape. Essentially, a large right ventricle is a bad thing, and that's what they want to avoid. However, LV dysfunction is also common. LV ejection fraction is often preserved, yet they can still go into heart failure with preserved left ventricular ejection fraction. And so a big question that the cardiologists have been asking, since a lot of studies have focused on the right ventricle, is what about the left ventricle? And so we actually made a cardiac atlas of 120 patients with repaired Tetrology of Fallot to look at the left ventricle. And our cardiology colleagues believe that this kind of information could give them better assessments of right and left ventricular dysfunction and improve understanding of patient outcomes. So first of all, what we did is we looked at these adult patients. Some of them were still minors, but they weren't infants and toddlers. They were teenagers to adults. And we looked at them by resolving the shapes of their left ventricles using the MESA atlas. So this is the atlas that I just showed you. This is the closest we have to an atlas of normal ventricular shape. And so the first thing you see, these are the modes of variation, the top 20 modes of variation on the MESA atlas of the tetralogy patients. And you can see that aside from the very first mode, which actually looks pretty normal, so it has a mean of close to 0 and sort of a standard deviation of about 1. This is on a normalized, this is a z-score. Aside from the very first mode, every other mode is statistically significantly different from normal. Okay? So left ventricular shape in just about every aspect of left ventricular shape you could think of is significantly abnormal. In some cases, the distributions are very wide. In many cases, the means are far from, they're 2 standard deviations away from the mean of the asymptomatic population. Interestingly, the variation isn't so wide, but the means are even further away from normal if you look at the wall motion. So every single of the top 20 MESA wall motion modes is abnormal for the tetralogy population. Okay? And you can see in some cases, the mean of the tetralogy population is nearly 4 standard deviations away from the normal adult population. And so this is the problem that the pediatric cardiologists have. Often they're seeing adults who they've at an age with a disease that previously would never have lived. So they're seeing a 40-year-old or a 50-year-old with a repaired defect they've never seen. Nobody's ever seen anyone older than that. And they have shape and function characteristics that are off the scale. So just to look at one that was particularly abnormal was end-diastolic mode shape mode 9, and you can see interestingly, so here's the in red this is the distribution of the MESA population of this mode, and this is where the blue shows where the tetralogy cohort fits in this mode. And the interesting thing is it almost looks like they fit on the better side, the more conical, less spherical side of this distribution. Yet this particular shape mode explains a much greater fraction of the variation in the systolic wall motion than the others. So there's something unusual about this shape mode, and the proportional reduction of error in all of these systolic shape modes, you can see that a lot of them is quite large. Okay? So there's something about this abnormality in shape, which a positive value of which seems to actually be a good shape. That explains abnormalities in function, most of which are dysfunctional metrics of dysfunction. And so we don't really know what this means, but the cardiologists are very interested in using this index as a way of seeing if they can predict outcomes for some of these long-term results. But there's an issue here. the MESA shape modes, all of them put together, all top 20, don't really account for much of the shape variation in the left ventricle of the tetralogy patients. In fact, it's only about 12%. So furthermore, the variation in shape of the tetralogy patients is actually 73% greater than the variation in shape in the asymptomatic population, which isn't surprising. But the result of this is that for understanding variation within the tetralogy, the MESA atlas, the normal atlas, is a good way of seeing what's abnormal about these, on average, about the tetralogy ventricular shape. But it isn't very good at understanding the variation within that group. Here we see the principal modes of the tetralogy atlas. Here's mode 1. And mode 1, you can see here, is mostly reflecting a difference in size of the ventricle, even though we normalize for the height of the patient. And this mode actually explains 20% of the total shape variation, 26% of the total shape variation of these patients, compared with just 12% of the shape variation that's explained by all 20 of the MESA modes put together. In other words, here's a number that doesn't come out of analysis of the normal adult left ventricle that has a lot to do with the variation of the tetralogy patients. And so this again could be clinically important because it means that the type of metrics that we're used to looking at and understanding the variation of in the adult population may not be capturing the important properties of the shape of the left ventricle that vary within the congenital heart disease population. Just looking a little bit, you can see that there were some fairly highly statistically significant and strong correlations of this mode 1 with the size of the left ventricle, but not with the ejection fraction. In fact, there's a slightly negative correlation there, and also with QRS duration, which is an indicator of risk for arrhythmia. We could also then see, how this parameter is explained by other clinical variables, and you can see that compared, say, with the MESA mode, a much greater fraction of the variation of these clinical variables will explain the variation in this shape mode than the MESA atlas parameters do. So I probably should keep moving, but quickly, in trying to understand how patients respond to therapy and what their outcomes are going to be, investigators have tended to use the size of the right ventricle, a change in the size of the right ventricle, a change in the right ventricular ejection fraction, or a change in the left ventricular ejection fraction as metrics of how well the patients are doing. So if their right ventricular end diastolic volume increases, if their right ventricular ejection fraction decreases, or the left ventricular ejection fraction decreases over time, these are thought to be prognostic of potential future heart failure and indicators that some sort of intervention, such as pulmonary valve replacement, may be warranted. So what we did is we looked at the shape mode parameter and against the changes in these clinical measurements over a 2-year follow-up. So nothing happened in these patients. We're doing a study now where we look at the before and after valve replacement. This is just the result of approximately 2-year follow-up, what changed, to see whether the adverse changes might be explained by, or might be captured by the this shape mode. And you can see that in each case, the parameter that best predicted the predictive variable here that best predicted the change in the response variable as the column heading here, the highest one is shown in bold. And you can see that quite a few of them, for a couple of them, like the change in left ventricular end systolic volume, of right ventricular end systolic volume, and although it's not the highest, of right ventricular end diastolic volume, and change in left ventricular ejection fraction, were actually captured by this single shape mode. So this was a better predictor of the 2-year change in these 3 variables than any other variable, and one of the best predictors for this variable. So our clinical colleagues are actually quite excited about the possibility that this could help them make earlier determinations for intervention. So finally, those of you who do cardiac modeling probably don't just do statistical modeling of shape. You're actually interested in models of shape because you can then use it for models of mechanics or electrophysiology or metabolism. We actually have a similar interest, and we've started to wonder whether we could use models of mechanics to create essentially atlases of mechanics. So one way we could do this is we could do inverse analysis and patient-specific models of every patient, but that tends to require more information than is available. So the strategy we're using is sort of the opposite. What we do is we optimize just the mean shape to find the mean material parameters that will predict the mean changes in end systolic function. So we essentially, for the mean end diastolic shape, find the material parameters for the mean hemodynamics that will predict the mean end systolic shape. Having done that, we then apply those parameters to every patient and then use that to predict what the end systolic shape would have been if the hemodynamics, if unknown, or the known hemodynamics and the material properties were normal, or at least were average. So that way, if the predicted end systolic motion was greater, it suggests that the material properties weren't as great as average, and if they were less, it suggests they were greater than average. So in this way, we can use the discrepancy between a model prediction and observation as a surrogate for a material parameter such as contractility. So this is a strategy that we're trying now. We have a similar strategy for electrophysiology, since a lot of these patients tend to have conduction block and prolonged QRS intervals and increased risk of sudden death. But I wanted to use the rest of my time to talk about a completely different, although scientifically related problem, which is the cellular mechanisms that underlie changes in ventricular shape during ventricular remodeling, and particularly the role of mechanics itself in regulating that, by telling you a little bit about some of our work on modeling cardiac mechanotransduction pathways. Now, we actually have models for mechanotransduction both in cardiac myocytes and cardiac fibroblasts, and we're working on models on the paracrine communication between them. But today what I'll show you is models that Kyle and Philip made of myocyte mechanosignaling, where one of the main outputs of that is myocyte hypertrophy and growth. So there's the model. I realize you can't see it or read it, but I'll try and take you through it. The way we built this is we had 172 experimental papers that defined this network. So everything you see in this network was our interpretation of a more or less consensus of the literature. Okay. You can see that at the top we have stretch, and then we have some mechanosensitive or nodes or mechanoresponsive nodes. So stretch affects 9 nodes directly. 5 of them we call mechanosensors because there's evidence that stretch itself directly affects the molecular properties of the angiotensin receptor, the L-type calcium channel, TRP channels, integrins, and dystroglycans. There's another 4 mechanosensitive nodes where we know there's a fairly immediate response to a mechanical stimulus in the sodium proton antiporter, angiotensin 2 secretion, endothelin 1 receptor, and GP130, but there isn't direct evidence of a direct mechanical effect on these nodes, so we call these mechanoresponsive nodes. The stretch feeds into all 9 of these nodes. Then there's 65 signaling molecules. They're mostly sort of well characterized. We've got MAP kinases, we've got IP3, PI3 kinase signaling, calcium CAM kinase signaling, integrin signaling through FAC and SAC, RAS, and some cytoskeletal signaling mediated by alpha-actinin, Titin, FHL1, ROC. So these are actually, for those who study this stuff, these are well known. There's nothing sort of exciting and novel in here. At the bottom, we have 11 transcriptional regulators, transcription factors, that bind to DNA and affect the expression of genes. And then we have 10 phenotypic outputs. So these are the sort of things that people measure in their papers, such as protein synthesis, cell size, gene expression of atrial natriuretic peptide and B-type natriuretic peptide gene, circuit gene, alpha-myosin heavy chain, beta-myosin heavy chain, alpha-actin and connexin 43 and angiotensinogen. So these are all the kind of markers, responses to stretch that are well characterized, many of which are associated with hypertrophy and that appear in enough papers that we could formulate this network model. In all, we ended up with 94 nodes and 125 reactions. And by now you're thinking, okay, you're thinking what von Neumann was reported to say, which was with 4 parameters I can fit an elephant, and with 5 I can make him wiggle his trunk. And the funny thing is, actually, there's this group in Germany that actually did manage to do this, although, you know, in fairness, they had 4 complex parameters. Right? But then with 5 complex parameters, they did actually make him wiggle his trunk. Okay? So there is a problem here that you may have recognized, and that is that this isn't an elephant. Right? This is an elephant. And I was about this far away when I took this picture in South Africa of this baby elephant. And the mom was like that far away. So this is the problem, and this is the reason why parsimony in biological modeling is only going to get you so far. If the previous diagram is your idea of an elephant, you're good. But this is an elephant. Okay? And that's why we don't have the luxury of reducing this model to the few parameters that are needed to explain the key idea that someone thinks is the important idea about mechanotransduction, because we don't even know what the important idea about mechanotransduction is. We don't even know if the most important thing about mechanotransduction is even in our model. We just took what 172 papers thought might be the most important thing and put it in. Okay? So how do we validate? Well, we were able to validate the model by qualitatively, and I should say something about the parameters. So actually the parameters of this model were not really free. We use a normalized Hill type function with a Hill coefficient of 1.4, a time constant of 30 seconds for every single node in the network. Okay? So really there weren't too many free parameters other than the magnitude of the stretch and our interpretation of the output as to what these meant. But we set a threshold for the outputs of 5%, and then compared input-output predictions, and actually got out of 61 results in 54 papers that we'd set aside that weren't used to make the model in the first place. We were 100% correct and just did this output of the model go up or down. We weren't quite as successful when it came to studies where intermediates in the model were inhibited. So here you can see sometimes the model predicted no change when there was a change, like a downregulation. Sometimes we predicted a downregulation when there was an upregulation. But overall, we still got 73 out of 111 predictions correct. And if we didn't get some wrong, there'd be no scope for learning from this model anyway. So this is a good thing. Okay. Okay. How did these accuracy conclusions depend on variation? So we randomly varied parameters in the model, the height of the Hill curve, the weight on each node, the Hill coefficient, and the EC50 of the functions, all of which were kept constant in the model results I'll show you, but we randomly varied all of them to see how our estimates of accuracy changed. And you can see we really had to vary them a lot before we significantly departed from these kind of 80% accuracy estimates. So in other words, this conclusion of the model accuracy we think is reasonably robust. We did use the model to compare pairwise effects of inhibiting every pair of nodes in the network on markers of disease. And the one marker of disease that is a particularly useful one is one that was discovered by my colleague Alan Maisel at UCSD is plasma BNP. So the expression of BNP either in the tissue or can be detected as a blood biomarker of the B-type natriuretic peptide is a sensitive indicator of the heart being overstressed and being at risk of heart failure. So we could use a decrease in BNP expression when you inhibited a node okay, or two nodes, as a sort of a readout of where are we getting synergistic effects of a paired inhibition. And so what you see here is the decrease in BNP expression induced by the greater of the two of the pair, and this is the additional increment you get by doing both. Okay? And then the ones that are in yellow and red are the ones where one of the pairs was the angiotensin type 1 receptor, and the other, and the yellow are the ones where one of the pairs was cyclic GMP. And I'll tell you why in a second. And essentially, one of the high hits up here in this corner of the graph was inhibiting the angiotensin receptor and increasing cyclic GMP. And the interesting thing about this is what happens when you inhibit neprilysin, which is what another drug called sacubitril does. The really interesting thing is that the angiotensin receptor blocker losartan and the neprilysin inhibitor sacubitril is a drug, the combination drug that was recently approved, seems to work well for heart failure with preserved ejection fraction. Is called Entresto. But at the time we did this, nobody had actually ever tried Entresto on stress signaling experiment, at least on looking at these hypertrophic markers. So this is what happens when we inhibit, when we give valsartan, the angiotensin receptor, and we increase cyclic GMP to simulate the effect of inhibiting neprilysin. You can see we get an additive, significant additive effect of blocking both, of both drugs. And as I said, that was a prediction that wasn't actually in the literature, even though it's an approved drug. But actually, there was a study that came out the year after we published this paper that did actually confirm that some of these hypertrophic markers, such as cardiomyocyte size, were actually significantly more inhibited with Entresto than with valsartan. Okay, so that's a published paper. Going on from here, what we wanted to do was, this is the same model, but what we wanted to do was use the fact that we had these transcriptional regulators to create a model of transcriptional regulation of downstream genes. And so we ended up finding 784 putative target genes of these 11 transcriptional regulators. I should say of these, only 293 have been experimentally validated either in the mouse or in the heart. Okay, I'll explain that in a second, why that's important. It's obvious why it's important in the heart. I'll explain why in the mouse in a minute. That resulted in an additional 921 reactions. Again, they just come from our reading of the literature, which required finding an additional 115 references, of which 45 were heart or myocyte studies. Then these 14 genes here, out of those 784, actually coded for proteins that are nodes in the network itself. Okay? So I've shown these because we also created translation reactions whereby the expression of this gene was assumed to feed back directly into the activity of the node. So we just assumed that if the gene was induced, the protein was upregulated, and if the gene was suppressed, the protein was downregulated. There was no more complex post-translational model. Okay? But there was this level of feedback from these 14 additional genes. So here's what the model, sort of thing the model does now. So at 0 minutes, nothing's happening. And then you can see by 2 minutes, the receptors have been activated, some of the second messages are turned on, and we're starting to get phosphorylation of MAP kinases and activation of HDAC and PKC. And then by 30 minutes, now the transcription factors have been activated or suppressed, and you're even starting to see some immediate early genes like FOS and JUN be turned on. And then by 4 hours, we're now starting to get, some of them are actually going down again, and we're getting some maximal responses of some genes, and most of the genes are getting switched on. And actually this goes on for quite a while because of the feedback. Before it reaches steady state, it takes over a day. But I showed you 4 hours because that's the time point, and 30 minutes because those are the time points we did our experiments in. So this is how we did the experiments. This is supposed to be a picture of a silicon elastomer that we microfabricate with channels that are 10 microns wide and 10 microns apart and 5 microns deep. And if you take mouse neonatal ventricular myocytes and you plate them on, if you coat this with an extracellular matrix protein like laminin, and you take mouse ventricular neonatal myocytes and you coat them on the surface, they sort of nestle into these channels and they become aligned and elongated. They sort of roll over the top and are able to form junctions side to side with neighboring myocytes. And they're also able to form end-to-end junctions. And we've previously shown that this kind of microscale engineering gives you about the morphology, the size and the coupling, myocyte to myocyte coupling of in vitro that you would see in the adult mouse heart. Okay? Instead of just being randomly organized as if it was on an unpatterned substrate. What we can then do is take this membrane, and we actually do this first before we put the cells in, but this membrane can go into a device that's designed to stretch the membrane, and the way it does that is it just has a screw, this indenter, it actually happens to be elliptical, and then it has a screw, and you screw this screw down over the membrane, which is held in place by an O-ring, and as you displace the indenter, it stretches the membrane. But because the membrane and the indenter are elliptical, the displacement is constant, but the denominator, the length of the portion that's being displaced, is not. So the strain is anisotropic. The result is that in the long axis of the ellipse, the strain that we use is about 4%, about 3 to 4%, whereas the strain in the short axis, where the denominator is shorter, is about 14%. Okay? And so depending on whether you align the grooves in the membrane this way or this way will determine on whether the predominant strain that the myocytes feel is transverse to their myofilament axis or parallel to their myofilament axis. Okay? And the important thing about that is that both of these strains are typical of what happens in vivo simultaneously at both, at the same time. So myocytes are in the wall are being stretched both longitudinally along their axis and transversely during filling, and they're shortening in 2 directions during ejection. Okay? So these are not unphysiological strains, but one is longitudinal dominant, the other is transverse dominant. And so this is what happens if you then take those cells, isolate RNA after 30 minutes or 4 hours, and then use RNA-seq to measure the change in expression of the messenger RNA as a result of this 30 minutes or 4 hours of transverse or longitudinal stretch. Interestingly, the very first thing that happens, the biggest response that happens, is 30 minutes after transverse stretch. You actually get a sort of transient induction of genes. Okay? These are mostly called immediate early genes. But notice they then go down by 4 hours. Okay? Whereas less happens with 30 minutes of longitudinal strain, but by 4 hours of longitudinal strain, you get a large upregulation of genes, a total of 795, compared with only 35. Right? So a huge difference between longitudinal strain and transverse strain on the induction of genes in as little as 30 minutes, and especially by 4 hours. Okay? Very big differences. If we were just to do some sort of naive clustering of these genes by annotations in the gene ontology that they have in common, you would see many of the biological processes are ones that you'd expect to see, like regulation of cell growth, cardiovascular system development, muscle cell proliferation, signal transduction, protein phosphorylation, muscle contraction, adhesion, cytoskeletal organization. These, particularly with the 4-hour longitudinal stretch where you get most of the induction, these GO categories are also enriched in the nodes in our network model itself. Okay? There are some additional genes actually that are downregulated that are not in the model and not in the literature, and they're actually ion channels, so particularly potassium channels. And it's interesting that the literature seems to be biased in favor of genes that go up rather than genes that go down. You can see the same for if you look at molecular annotations or pathway annotations. So MAP kinases comes up, and obviously we had MAP kinases in the model. But you also get TGF-beta signaling, Hippo, TNF-alpha, they aren't in the model. So these are categories where we could be extending the model. So I wanted to talk a little bit about the validation criteria. These are the sort of the details that I tend to gloss over that I think are important for today. And that is exactly, when we come to validate this, how valid is the data that we're using to validate with? This is the big problem, right? How do we know that this gene did go up or didn't? How do we know this gene should have been included in the model or not? How do we know whether we should be comparing this gene or not? Okay? Because we don't really have very stringent criteria for determining which genes should be in the model, or at least if we do, we don't get very many. Okay? So we have a choice. We can get a lot of genes that we have some reason to believe they're there, or relatively few genes where we have very good reason to believe they should be there. Okay? So we actually have three different criteria. The first criterion is that transcriptional regulation was identified experimentally in experiments that actually used cardiac cells. Okay? They weren't all mouse. Often these are done in rats and sometimes in other species. That's our most preferred criterion, but there aren't actually that many genes that fall into this category. The second criteria is that transcriptional regulation has been identified from experiments using mice or rats, okay? Not necessarily heart cells. Okay? So we confirmed that this gene is regulated by this transcription factor in a mouse or a rat cell, but not necessarily a mouse or rat heart cell. Okay? Finally, transcriptional regulation has been predicted using promoter binding sites from published ChIP-seq data. So this is an assay for chromatin immunoprecipitation assay for identifying targets of specific transcription factors on the genome, and a bioinformatic analysis called iRegulon. So this is an experimentally based bioinformatic prediction, but nobody then went and inhibited that transcription factor and confirmed that that gene was actually not expressed, let alone in a heart cell in a mouse. Okay? So depending on which criteria we use, we can be even more stringent. We could require all three, but then we only get about a dozen genes, and we sort of lose statistical power a bit to make conclusions. The model looks good, but it's very similar to the model I've already showed you, where we just had a few selected phenotypic outputs. If we allow either criterion 1 or 2, there are 293 genes. If we allow either criterion 1, 2, or 3, then we get 784. And of these are all genes that are detected in our experiment, but of them, only 42 of these were differentially expressed using a false discovery rate of less than 0.05, or 129 were differentially expressed, meaning up or down regulated significantly by a false discovery rate of less than 0.05. So false discovery rate is just a common correction for multiple comparisons that's used in genome scale data, and it's the particular algorithm is called the Benjamini-Hochmuth algorithm. It essentially takes the most significant p-value and divides that by the total number of genes, and then the next most significant and divides that by the total number minus 1, and so forth. So then the next question is, how are we going to establish thresholds? I didn't mention, but whereas all the signaling aspect of the model is normalized, the gene expression, we tried this for a long time and we just couldn't make it work. And the real reason is because nobody measures normalized gene expression. Nobody knows what maximal gene expression for any experiment is. Everyone normalizes gene expression to the baseline, and the baseline is highly variable between different conditions. And so we finally gave up on trying to use this normalized strategy for predicting the gene expression, and we instead used a strategy where we actually quantitatively use mass action. So we use a normalized transcription factor activity to activate mRNA synthesis. And then we compute mRNA degradation using databases of mRNA half-lives. So people know the decay times for mRNAs, and so we could put the rate of decrease of mRNA into the model quantitatively. Then all we need to do was solve for the rate of transcription for each gene, and we used that by assuming that the baseline condition was at steady state. Okay? This allowed us to make a quantitative prediction of gene expression, which then made it easier for us to compare with experiments. Okay? And we really only did this to compare with experiment. We still had a problem. Experimentally, conclusions of whether a gene changed or not aren't based on the magnitude of the change. They're based on the probability, right? They're based on the false discovery rate. So here we're plotting the fold change on a log2 scale of gene expression up or down in our data for the large data set and the smaller data set of the p-value of alpha, where these dotted lines represent a false discovery rate of 0.05 for up or down. And fortunately, you can see that sort of in this low expression range, this curve is fairly tight. And so if we use this envelope, we can actually estimate a threshold for upregulation, a quantitative threshold for upregulation that's consistent with a p-value or a false discovery rate of 0.05, and similarly for downregulation. Essentially, that number is about 0.5 on a log2 scale, slightly higher for up or down. Okay? So that's how we determined our threshold. Okay? Here, if we plot just the absolute expression of the genes that are measured versus the prediction of the model, you can see overall the model does pretty well. But this is a bit misleading because this is the full dynamic range of all the genes, which we've mostly accounted for in the baseline. The change in expression is not nearly this wide of a dynamic range. Here's the errors for the log2 fold change. And you can see that more than 90% of our predictions fall within plus or minus 1 on a log2 scale. On the other hand, so do a lot of the changes in genes that occur in these experiments on these relative short time scales. These are just organized by the different transcriptional regulators. Okay, so here's now one way of looking at the model predictions. We can look at the log2 fold change of the model prediction versus the log10 p-value of the data, and that will partition our data into 9 groups. Those, for example, the big group where the gene didn't change in the model or the data. Here, the gene went up in the model and the data. Here, the one gene went down in the model and the data. And then here are some of the incorrect predictions of the model. So if we look at this for the 293 validated genes, then we get a 77% overall accuracy. I think I meant to look at this first. But there's something interesting about this. So there's a sort of a 3-way contingency table you could make, basically what I just showed you. Or we could break it down into a more conventional 2-way contingency table by either discriminating the data as whether they're differentially expressed or not, or from those that are differentially expressed, whether they went up or down. You can see by the time you take those that are differentially expressed in this group and you look at those that were predicted by the model, whether they went up or down, now the accuracy is really good, but the numbers are small. Here, on the other hand, the numbers are much bigger, but notice that there's a difference between the true positive rate, which is only around 30%, and the true negative rate, which is about 84%. This has a lot to do with the threshold we use for determining whether the gene changed in the data, and I'll come back to that. Here's the same thing for the 784 genes. You might think that maybe this isn't going to be as good because, after all, most of these genes are less stringently validated. But interestingly, the accuracy is pretty similar, about 72%, not quite as high, but quite similar. And now we get larger numbers to start to do some statistics on. Here again, though, same thing. The true positive rate is significantly lower than the true negative rate. Okay? Although if you just look at up-down, the model does pretty well and the numbers are small. So we started to think about this. What does it mean that the true positive rate is low and the true negative rate is high? What it really means is that the threshold in the model is we're predicting a lot of genes to change that didn't change in the data. But then the question is, how confident are we that the genes in the data didn't actually change? And the question, that's determined not by the false discovery rate, not by alpha, but by beta. So what is beta? What is the power when we set a false discovery rate or alpha of 0.05? It's terrible. Okay? And this is true of pretty much every genome scale data set you'll ever see. Okay? Except for those few sort of aficionados who know this well and will do 10 or more very expensive replicates of this type of experiment. But almost every paper you see published will use somewhere between 2 and at most 5 replicates. Okay? So because we used 3 replicates, the power was understandably miserable. Right? In other words, beta was 0.467. It was 9 times higher than alpha. So the confidence that we could say that a gene changed is 9 times greater than the confidence we can say that a gene didn't change. So maybe this is why our true positive rate and true negative rates don't match. So there's different ways you could sort of explore this, but one simple thing is you say, well, I could change alpha to the point where alpha and beta were equal. At what point would we, instead of stringently being able to conclude that the gene was differentially expressed, at what point would we be able to conclude with equal confidence that a gene was or was not? And it turns out, if you just sort of figure it out, that the value is 0.21, okay? Not too many people are going to accept your paper if you use a p-value of 0.21. But if you wanted your power and the risk of a type 1 error and a type 2 error to be the same, that's the number you should use. I'm not suggesting we use it, but it's just kind of interesting, because then what we can do is we can go back and say, how would these numbers have changed if we had done this? And so this just shows, so here we are on the ROC curve when we use a false discovery rate of 0.05. Here's where we are when we use a false discovery rate of 0.21. And none of this is really surprising. It's just that you never hear anyone, I don't think in modeling or in this type of experimental work, acknowledge this limitation of these data. I think finally I'll just tell you a quick story about how we actually make this model useful, because I know my time's running out. One thing we could do is ask, well, what was it at the top of the network that was most sensitive to transverse stretch versus longitudinal stretch? So if we just inhibit one node at a time, whether you inhibit angiotensin or the receptor, sort of the equivalent the same. Interesting, how big of an effect do you have on those genes that are specifically induced by transverse stretch? And the biggest hit comes with angiotensin, and then with endothelin-1, then with integrins, and then you go down the list. Okay? And so we then said, well, what if we blocked angiotensin and endothelin-1 versus randomly taking 3 of the others in here, and the easy ones to choose were ion channels. Okay? So we predicted that there'd be an overlap, but a statistically significantly greater effect on inhibition by inhibiting the angiotensin receptor and the endothelin-1 receptor on those genes induced by transverse stretch than if we use these 3 ion channel blockers. Okay? So this was a prediction which we could then test experimentally. Here are the experimental results, here are the predictions, and I won't take you through them all, but essentially for these 6 genes that were predicted by the model, one of them is a positive control, or negative control, there's actually pretty good quantitative and qualitative agreement between the predictions of this analysis and the experiment. And so we ended up, by this and some other studies, concluding that this sort of sub-network of the model involving angiotensin and endothelin and its downstream signaling pathways was as the main mediator of the response to transverse stretch in our experiments, whereas the entire network is essentially responsible for the response to longitudinal stretch. So in summary, population variations can be useful for identifying novel predictors of risk or clinical outcomes, and underpowered genome scale data can influence the conclusions of model validation comparisons. So with that, I'll thank you very much for your time.
Thank you, Andrew. It's nice to see the complete span of scales in cardiac modelling, from the very big to the very small. Have we got some questions? Yes, Caroline.
Thank you for the talk. In your shape
Could you
models
speak
of
quite loudly, because it's... Thank you.
In your shape models of tetralogy, did you consider the presence and the location of the VSD patch?
So that is a very good question. We did not specifically. However, what we are doing, at least for the arrhythmia studies, is we're looking at exactly where the patch is and the size of the scar, because that seems to be an important determinant of the conduction delays. So it didn't enter the models, but we do have that information that we can superimpose on the models. And it's a really good question. It's very, very important, particularly for the arrhythmias. Yeah. Syria?
In your stretch experiment, did you paste the tissue?
No.
So that the condition corresponds to diastolic overload?
Yes. I mean, these cells are spontaneously active. We've never found much of a need to try and cycle stretch to I think of the load that we impose in vivo is more like a chronic hemodynamic load, not like a beat-to-beat load, Because even if we could pace, all the cells would have to be synchronized with the pacing, and so no, we don't pace, but they are spontaneously active, and it's a static stretch. One of the reasons we only go out to 4 hours is because after about 6 hours, the myocytes have actually added enough sarcomeres in series that essentially don't feel that strain anymore. And I didn't mention it, but we actually include that feedback in the model. So one of the parameters of the model was cell area, which we can take the square root of to convert to cell length, and we can then have that subtract from the stretch, such that the stretch stimulus in our simulation is actually a transient that decays away, because we think that's what's actually happening in vitro. Edmund?
Andrew, in the context of uncertainty quantification, I'm thinking about your network modeling. So you've got a very carefully curated network model for the signaling. But I suppose, nevertheless, we still have to consider the possibility that there are missing edges, that there are connections that are missing. So I'm wondering if you have a sense of how one goes about assessing whether some of the insights that you get from your modeling, how sensitive they are to that sort of structural uncertainty in the signaling network.
Yeah, well, not only do I agree with you that that's almost undoubtedly the case. In fact, we do know of other pathways that are involved in similar mechanisms, such as hippo signaling, for example. One of the hopes is that sort of failure of prediction, particularly of sort of whole groups or classes of genes, like those downregulated potassium channels, will actually inform refinements to the model. We've really, until we publish the validation or complete the validation of this sort of next generation of the model, we've really tried to resist the temptation to change the model that it was based on, but the temptation keeps coming up because we keep finding discrepancies that can be explained by modifications in the model. We did, for example, find a specific modification of the model to the way that CAM kinase regulates HDAC. That if we make that one change, it actually improves the accuracy of the model. But we're trying to save that for a separate study so we don't change too many things at once. But yeah, I think actually one of the most exciting things about these large, sort of curated, largely qualitative models that are clearly missing a lot of information is their ability to help discover what's missing. The harder question is to what extent is that missing information confounding the uncertainty, which is your question, and I think the only way we can find out is by identifying what we think the missing information is and then repeating the validation analysis. So we're destined to repeat this validation like forever.
Andrew, really lovely talk.
Thank you, Bruce.
Really superficial question, but I've been fascinated by the fact that the response of the extracellular network to volume load as distinct from pressure load occurs over a very different time frame, and it's very different. It seems to me that the differences that you're seeing between lateral and longitudinal stretch coincide with that observation. So is that too simplistic a response to your data?
Well, what motivated this experiment in the first place was some of our continuum models where we did actually assume that transverse and longitudinal stretch were drivers of lengthening versus thickening of myocytes, and that does give rise in those quantitative, in those empirical models or continuum models. It does mean that if you create an aortic stenosis, you get a concentric hypertrophy, and if you create a mitral valve regurgitation, you get a dilated eccentric hypertrophy, and the time courses are different and they are consistent with the experimental observations. But the big problem here is the time courses for these models are still too short for us to really extrapolate to what will happen on the time scale of sort of weeks of remodeling. It actually takes days for this model to reach steady state, so it's not like we're way out of the ballpark, but there's a whole sort of second window of post-translational modifications and additional changes that I think we'll need to get before we can really start to predict, the sort of physiological or pathophysiological scales of hypertrophy.
Great. Well, if there's no further questions, we're back in here at 11:30. Please look at the posters, and let's thank Andrew again.
Thank you.